Our next speaker is Professor Nathan Intrator, a professor of computer science and neuroscience at Tel Aviv University and the founder of Neurosteer.
Thank you very much. It's an honor to be here. So I've spoken a few times. I'm going to take us to biomarkers this time. And from the previous talk, I realized that we are a little bit late approaching FDA, but we just really frozen the hardware, a new version of the hardware recently, and I thought that it doesn't make sense to approach FDA before that. We are redoing now experiments that we've done with the old hardware. We are doing it with the new hardware. So what is it that we are really trying to do? We are really trying to replace the very old way to approach EEG or to approach brain scanning. fMRI is, of course, a totally different clinical device. And as I like to say, nobody's walking in the street with an FDA on their head. So it's obvious why we want to replace the FDA, but we want to replace the EEG, the current approach to EEG, because it's multi-electrode, because it's not really scalable, because it requires a technician to install, and because it requires a neurologist to look at the data. So what it is that we are bringing to the field, we are bringing a very small patch that is attached to very small electronics. Everything is wireless. That's on from the experience of the patient, from the experience of performing the test. We are bringing automatic kind of cloud-based tests that stimulate the patient or the person in real time and read the response of the patient. That reduces the need for a technician or for a clinician to perform the test. And most importantly, we are bringing novel mathematical analysis of the reduced data, because now it's a single EEG channel. So the novel mathematical analysis is what reduces the need for multiple channels. And this is really the biggest novelty of what we bring. For those of you who haven't seen this analogy, the analogy is our ability, each one of us ability to listen to an orchestra with a single ear and to decompose in our brain the full orchestra into the different musical instruments, okay? So this is, by the way, a very difficult signal processing, but because each one of us is performing that, then obviously it can be done, okay? And what is required is not just the frequency analysis of the signals, because frequency analysis is not going to distinguish between, say, a trumpet and a violin, but a much higher time frequency analysis of the data. And it turns out that a very similar problem exists here, because different functional neural networks in the brain have their specific timbre or color, just like musical instruments. And by listening, even with a single microphone or a single EEG channel, with advanced harmonic analysis, one can then distinguish between them, and this is what we are trying to do. So we are decomposing the brain activity, although we are listening with a single EEG channel. At this point, we are decomposing into 120 channels, but in the future, we will be decomposing into a much larger number of components. And the magic or the good news or etc. is that the fact that these components turn out to be related to some meaningful functional brain activities. What is our focus? So the current main focus is the top three, which are neurodegenerative and specifically cognitive decline as part of neurodegenerative. The goal is, of course, to provide this very affordable and automatic test at a very, very early stage, and to develop those tools that can detect at a very, very early stage that there is a problem, so that maybe Biogen's potential new drug, and other drugs can actually intervene at that very early stage, slow down the progression of the disease, etc. And of course, fMRI, which has been shown to provide some kind of early detection, cannot be used because it's very expensive, so it's not going to be applicable for that purpose. Our goal is to really bring it to the general physician's waiting room as a test that can be administered over there. Okay, so what are we doing in order to achieve all this? And today I'm going to try to focus on two biomarkers, two cognitive biomarkers that we have identified through quite a bit of work and data collection. And these two biomarkers are now kind of slowly being prepared to be submitted to the FDA as potential biomarkers for what we are trying to achieve. It's going to be VC9 and VC6, and I'm going to talk a little bit about them. So it's not enough to talk about a biomarker when you're talking about the brain. It's important to see what it is that you stimulate the brain in order to read a change in a certain biomarker in order to deduce something about brain activity. So the NBAC, which I'll mention, I'll list a little bit in the next slide. The NBAC is a very standard test to test working memory, and we are doing various types of NBACs, and the musical detection. Music, by the way, is a very strong stimulant to the brain. It makes the test much more pleasant to subjects, and does not require language knowledge, and we use music quite a bit. And it turns out that we can read various indications about resting state, default mode network activity of patients, and it also turns out that this is very, very relevant in very early stages of neurodegenerative disorders. For example, there's association between beta amyloid plaque and changes in default mode network, and this is something that we are exploiting quite a bit. So a word about NBAC, I believe everybody knows, but the point is that this center line indicates that it has been shown to be a very good predictor for MCI. So this is an old study. We have now many more patients and subjects, as you'll see soon, but the point is we first start with healthy subjects. We go to a validated, well-published NBAC test, and then we correlate ourselves, so kind of like a 510K predicate, if you want, and then we correlate our findings to the studied performance, and that's what we are doing here. So for this biomarker, we are now starting to get some results, and let's look at them. So in NBAC, there is zero-back, which is kind of the simplest one. You hear that musical instrument. One-back is you have to click if the current musical instrument is the same as the previous musical instrument. In two-back, you have to click if the current musical instrument is similar not to the previous one, but to the one before previous one, and then three as well. So clearly, the cognitive challenge is going up as the numbers go up. And what we see with this VC9 biomarker is that we see an increase in certain activity of that biomarker as we increase the cognitive challenge, again with healthy subjects. Note here that there's no effect for the zero one. This is for a numeric NBAC. When we go to musical NBAC, the picture changes a little bit. This is what we have. So the first time that we listen to these musical instruments or to these musical notes, we have an increase in activity. When we go up later, we get that increase that we had before. So that's for the NBAC. For the detection task, Again, we have to basically detect something like an old ball detection. Same basic idea. And from here, we also get very nice results, namely that there's a significance in change between detection 1, detection 2. Again, two different challenging tasks of detection. Okay, so talking about one biomarker so far, I'm going to switch to the second biomarker soon. Very simple, very clear results, very clear kind of assessment of that biomarker. And now we are going to resting state, okay? So we're going to be testing that specific biomarker in a resting state situation, and we are trying to optimize the resting state. What is the stimulation in order to produce the most optimal resting state? We are looking at 4 different resting states right now. But with healthy subjects, that specific cognitive biomarker that we know has to do with working memory goes down when resting state kind of stimulation is applied to subjects. This turns out to be very different when looking at neurodegenerative cases. I'm going to skip describing where the resting state is and go to look at Parkinson's patients. This is the setup. You see the electrodes. This is the sensor. There's a little bit of electronics behind the ear, earphones to produce the stimulation, and our analysis. And if you remember how the NBEC looked before, now we see that it actually changes, and there's a reduction in the activity of VC9 as opposed to increase as the challenge increases. Okay, so that's already a very interesting change and a very measurable change that we can rank patients based on that change. I mentioned the resting state condition, and again, as I said, that in the case of Parkinsonians, for example, early Parkinsonians as well, the resting state the VC9 activity, that specific biomarker resting state, actually goes up instead of going down. Okay, so we see that because now we see a very clear difference between subtracting the activity during cognitive challenge and that activity during resting state, and we see a very strong difference. And as I mentioned, this enables us to rank patients because that significance is only being used for us to tell that something good is happening. What is really most important is to be able to rank a patient. And if we are talking about early tests that will be done in the future, then to compare that patient this year to the baseline that patient created the previous year in order to see whether there's been a change. And if we are talking about intervention and drug intervention or neurostimulation intervention, then the goal is to see whether there's been a change after such intervention. So you can see here that the healthy subject go into that positive difference, and the sick subject or the Parkinsonians mostly go into the negative subject. So talked about patients, talked about this VC. I'm going to switch to a few things that we've done just very recently that are kind of on the border between dealing with patients and dealing with healthy subjects. And the first is this project that we just did in San Francisco with the American College of Surgeons. The whole project was started by Professor Carla Pugh from Stanford. She's a surgeon and dealing with training surgeons to optimize their performance. And the whole idea was to quantify surgeons in order to be able in the future to be able to provide some kind of certification to their abilities, something that is done, for example, for pilots on a yearly basis. So each surgeon had then sensors on the fingers that were measuring the movement, accelerometers, a microphone, a camera. The camera was replaced with a camera somewhere else. Our sensor for cognitive assessment. We managed to collect over 200 surgeons within 3 days of the conference, which was quite exciting and also challenging because of electronic noise and whatnot. And here is how the project was going on. First, they were sitting here and again in a very noisy environment, sitting and performing automatically our cognitive assessment. Okay, so they had one of these monitors, they had earphones, they had a clicker in their hand to provide feedback, and they were performing automatically that cognitive assessment of ours. And then they went to perform an operation. Again, they had this sensors attached to their fingers, and they were performing a real operation in which the performance was actually tested at the end, the quality of what they were supposed to do. And we managed to collect this large number in very few days. By the way, the surgeons themselves kind of provided very nice feedback, both for the cognitive assessment, which they said was very difficult, which is kind of what we wanted to do. That's going to be a different, of course, assessment than we provide to Parkinsonians or to neurodegenerative cases, but we applied that. And if we look at the results, we see the same results. Notice we are looking at VC6 here, which is another cognitive biomarkers of ours. We see kind of the same story that 0 NBEC, 1 NBEC, the performance is very, very clear. Now, what was interesting here is that some of the surgeons were retired surgeons who haven't performed the surgery for quite a long time, that they were over 67. So if we look at the results of the non-retired surgeons, this is the results that we see. But when we look at the results of the retired surgeons, this is the result that we see. And again, now we can see that their cognitive behavior, just like in the case of early Parkinsonians, we can see a change in the activity of those biomarkers, of this biomarker in this case. Okay, so healthy, but some of them were close to 80 years old, so potentially with some kind of specific disorders. I'm going to talk quickly, actually not so quickly, about response to TDCS, to transcranial stimulation, DC stimulation. This is work we are doing on comatose and minimal consciousness patients. This is work, this specific results are from Jerusalem, from Israel, from Herzog Institute. First looking at alpha as a biomarker for change in stimulation. These patients undergo 12 sessions of a stimulation, and apparently they do show a change, a big change in their ability. I'll discuss that a little bit. So alpha is kind of in the literature suggested as a potential comparison, and that enables us to compare our results or our biomarker, the sensitivity of our biomarker to alpha. Here are some of our BRAFs, which are the brain activity features, which I kind of used to talk about in the past, but today only talking about biomarkers. Here they are pre and post for these 4 subjects that we used. And now comes the question, well, what can the doctor learn when a specific subject is being provided? And this is something very, very interesting. So let me explain to you what we see here. We see the left axis is basically the delta between the activity of a specific biomarker and the activity before the stimulation and the activity after the stimulation. So we see that usually it's positive or above zero for most of the biomarkers and most of the patients. We have now 5 patients here. We have one patient that the result is totally different, okay? And of course, that is a very clear indication that something is different in this patient. It turns out that the injury of that specific patient is much larger. There's several issues there that may explain, but the simple bottom line is that the resting activity of that patient is extremely high, much higher than would be expected at any normal situation. I mention it because we see that in strong Parkinsonians all the time. We see that in gait freeze or people that are close to gait freeze. So we see that quite a bit. That's not something that's a frontotemporal problem that we see quite a bit. And what happened in his case is that the stimulation actually reduced the resting activity and actually improved the result as a result of this. But here we basically see a negative number, and that's quite interesting. By the way, this is a coma recovery scale, so we can apply that to each part of the coma recovery scale. So you see that in auditory, you see that in leg stretch, you see that in imposing pain, and you see that again with pain, you see that with visual stimulation. Okay, so having spoken about patients and serious patients, let me switch gears and finish with something much more exciting. And this is the marathon in New York that occurred a week ago in collaboration with Professor Stephen Lowry's, who is one of the top coma patients in the world, and we work with him in Liège. He's also a marathon runner, and he ran with his son the Marathon of New York. The initial plan was to run with 3 different EEG devices. So with a full montage EEG of 8 electrodes. With our sensor, you can see our sensor here on the forehead. So with our sensor on the forehead and with also EEG device that's in the ears. When he tried to run with the full montage, he gave up on it because it was okay when resting, but running produced very strong artifacts. And there was no chance that something useful will come out of it. So eventually they ran just with our device, which made us very worried because we were afraid that the device will fail with communication after a few hundred meters, and then he just came to the US in vain. But luckily that did not happen. And it turned out that we were able with these electrodes to deal with the sweat quite well, and we got a very good signal the whole time. In fact, they forgot to turn it off after the run, so they were connected for 7 hours straight to our device. We replaced the electrode one time in the middle just because we were too worried, and we wanted to be sure that the final part of the run will go well. So you can see them at different, the initial plan was to really go meet them and replace the electrode every 2 or 3 miles. But then when we saw that the signal was good, we were just running from one place to another, running via the subway in order to tell them everything is fine, continue to run, until we changed it one time. And as you can see, they ended up finishing the marathon with the device. We were relaying the information, the real-time interpreted brain activity via Facebook to the world, and there were several people watching it, so it was quite a bit of fun. And I'm going to just show 2 slides from that, but I would mention that the previous book that Stephen Lowry's wrote was about meditation. He is a good friend of the Dalai Lama, and he's analyzing two of the designated guys from the Dalai Lama, which is Mathieu Ricard and Lama Zopa. And I'm very thrilled that he switched from using 256 electrodes device to our device with these guys. And as part of this, he himself got into meditation, and we can see that in his run. And here is kind of one slide from the run. So that's basically how it was published on Facebook, and you can go to the video and replay the whole thing, about 4 hours of the run. So he himself, and now this is for those who saw my brain activity representation in the past, but basically blue is less active, red is more active. This is, by the way, VC6. This area is the executive area. This is VC6. VC9 is right here. This is VC9. So we can see that Stephen actually was meditating a large portion of the run, and it makes sense because meditation reduces brain activity, reduces consumption of oxygen, and of course enables a person to utilize the oxygen for other reasons. So here's the second one where he was getting into a deeper meditation. His son, who ran very easily but is less experienced in meditation, was not getting into meditation at all. There was a very short chunk of time where this was happening. So to summarize, what we are trying to do is to introduce a very small, affordable device that can provide early indication for serious neurodegenerative disorders, that can provide mass assessment of patients for early neurodegenerative disorders, can actually go home, can provide very good screening to pharmaceutical companies that are going to perform clinical trials. And sometimes when I hear people say Parkinson, I kind of think that we are like 30 years ago saying cancer, where the idea was there's a single cancer and a single drug for it, where obviously today we know that this is totally different. We, with our device, are seeing something like 5 different types of disorders when we are looking at, let's say, a Parkinson's patient. And I believe that being able to help pharmaceutical companies understand which of those populations their drug is useful and actually screen the patients with this kind of criteria, there's a better chance that the right people will go into the clinical trial, and potentially the drug will be approved. And as we well know, too many drugs have not been approved, and that may be one of the reasons. $3,000 for a 15-minute of fMRI is kind of too expensive to enable a very mass screening of patients for a specific trial. Okay, thank you very much. Yeah.
Does it work?
Yeah.
So considering the discussion before with FDA and your talk going to the market, are you planning to have or do you already have evidence from bigger studies when it comes to Parkinson's, and have you done any studies on Alzheimer? Because you only showed the healthy control data and nothing on Alzheimer, which also links to how much, if you want to target early, even preclinical phase in Alzheimer, just the correlation with one cognitive biomarker would be enough on your assumption or hypothesis.
Okay, so these are good questions. We have now close to 100 neurodegenerative disorder patients at different conditions. Parkinson, Alzheimer with depression, etc. Okay, we kind of used a lot of this data basically to develop our biomarkers. Okay, to really try to optimize those biomarkers, and this is now when we are beginning to feel comfortable that we have a biomarker that we can go to a next phase of clinical trials that will now validate that specific biomarker. So I showed here validation, say, on healthy subjects. That's beautiful and kind of shows that we are doing something, and it's still, even this is nicely publishable because there's no biomarker, for example, for NBEC that is as simple as a single EEG channel, okay? But that's not enough, of course, when you're looking at the elderly population. So this is now the ongoing work.
Have you tried to correlate or validate your data with functional MRIs? Question number one. And second, if you use both methods, would it improve the diagnostic value?
Yeah, so that's again a very good question. So we are trying to find those PIs or those people that have very nicely characterized patients, okay? So for example, I mentioned Stephen Lowry's, he's doing PET, CT, fMRI, 256 electrodes on his comatose patients, and that's an amazing characterization that we try to correlate with. With elderly population, yes, we are looking for those populations that had already fMRI done for various reasons, and is going, and then we can compare our stuff. Unfortunately, a lot of it I cannot talk because some of it is done with pharmaceutical companies, and I cannot provide too many details.
Thank you very much.
Thank you.